this topic, we're going to have a look at sex determination and sex-linked characters. So by the end of this topic, you should know how is sex determined, what is sex linkage, how is color blindness inherited, and how is hemophilia inherited. Now in humans, there are 23 pairs of chromosomes. We call 22 of those autosomes, and then the one pair we call sex chromosomes. In females, the two sex chromosomes appear the same and are called X chromosomes. In males, there is a single X chromosome, but the second one of the pair is smaller in size and shaped differently. This is the Y chromosome. Here you can see a scanning electron micrograph of the human X and Y chromosomes. Notice how much smaller the Y chromosome is. Now, unlike other features of an organism, sex is determined by chromosomes rather than genes. Females have two X chromosomes, and the gametes are the same in that they each contain a single X chromosome. What about males? Males have got an X and Y chromosome, so the gametes will have an X chromosome and a Y chromosome. So when fertilization occurs, you have the sperm fertilizing the egg. Which one determines the sex of the child? It's going to be the sperm. So in this case, since the sperm is containing the Y chromosome, the resulting child will be a boy. Okay, let's do a genetics cross. If you cross a female and male, always write parent, phenotype, genotype. So the phenotype is female and the genotype is XX, male, XY. You write down the gametes in circles, XX, XY, and then you use a Punnett square to do the cross. Your F1 generation will be XX and XY, so the phenotype will be female and male, and the ratio will be 1:1. This means that there's a 50% probability that the offspring will either be a girl or boy. Okay, let's look at sex linkage. The X chromosome contains many different genes. If it carries genes that are not present in the Y chromosome, these are called sex-linked genes. Examples include genes involved in color blindness and hemophilia. Okay, looking at color blindness, the gene for red-green vision is only found on the X chromosome. If we want to do a genetics cross, we write the sex-linked allele as a superscript of X because it's found on the X chromosome. So here is an example. If you want to write a normal person, you have X representing the X chromosome, capital N representing normal. Colorblind person, you'll have X with a superscript small n. So let's do a question. What will the resulting offspring be if you cross a female who has the carrier gene for color blindness with a normal male? Okay, your first step is to write the parents. So phenotype, genotype. A carrier female will be X capital N, X small n. Normal male will be X capital N, Y. Then what's the next step? You're going to write the gametes. So it'll be X capital N in a circle, X small n in a circle, X capital N in a circle, Y in a circle. And then you do the cross, and you do the cross using a Punnett square. So when you cross the male X capital N with the female X capital N, the result is X capital N, X capital N. And you do that for the rest. So you get your F1 generation. The offspring genotypes will be X capital N, X capital N, X capital N, Y, X capital N, X small n, X small n, Y. These phenotypes are normal female, normal male, carrier female, colorblind male. And what is the ratio? 25% chance of getting a normal female, 25% chance of getting a normal male, 25% chance of getting carrier female, and 25% chance of getting a colorblind male. So looking at the genotype and how the genotype influences the phenotype, you can see that only when the allele for color blindness is homozygous in females will the female be colorblind. What about in males? When the male has got an X, small n, Y, the male will be colorblind. Now, colorblindness is more common in men, where there are about 8% of men in the United Kingdom who are colorblind. Okay, let's have a look at hemophilia. Hemophilia is when the blood does not clot and it results in persistent bleeding. It can be potentially lethal if it's not treated. Although hemophiliac females are known, the condition is almost entirely found in males. One of the causes of hemophilia is that the person is unable to produce clotting factor 8. So the gene that codes for the blood clotting protein factor 8 is found on the X chromosome. Now, people with hemophilia can now be treated with factor 8 from blood donation, so they can lead nearly normal lives. So remember to write the alleles linked to the X chromosomes. So the capital letter represents the normal factor 8, whilst the allele that lacks this factor is small h. Both are written as superscripts of X because they're found on the X chromosome. Okay, let's do a question. What will the resulting offspring be if you cross a female who carries the gene for hemophilia with a normal male? So copy this question down and press pause and try it, and then check your answer. Okay, what you do is you write your parents' genotype and phenotype. So phenotype is carrier female, normal male. Genotype X, capital H, small h, X, capital H, Y. Then you write your gametes in circles. You use the Punnett square to do the cross, and you get your resulting F1 generation. So the genotype will be X capital H, X capital H, X capital HY, X capital H small h, X small h, X small hy. Phenotype, normal female, normal male, carrier female, hemophiliac male. And what is the ratio? It's 25% chance of getting a normal female, 25% chance of getting a normal male, 25% chance of getting a carrier female, and 25% chance of getting a hemophiliac male. Okay, let's do another question. What will the resulting offspring be if you cross a female who carries the gene for hemophilia with a hemophiliac male? Okay, write down your parents, carrier female, hemophiliac male. X capital H, X small h, X small hy, the gametes. You do the cross. Write down the F1 generation. Notice how if the female has got X small h, X small h, it will result in death. Okay, so the chances of a female being hemophiliac are very slim. As males can only obtain their Y chromosome from their father, the X chromosome comes from their mother. Since the allele for the non-production of factor 8 is linked to the X chromosome, males always inherit this disease from their mother. Since the mothers don't suffer from the disease, they must be heterozygous for the character, as you can see here. We call these females carriers because they carry the allele without showing any signs of the character in their phenotype. Let's do one more question. If a hemophiliac male had children with a normal female, would one of their sons be hemophiliac? So you write down the parents, phenotype and genotype. Normal female will be X capital H, X capital H. Hemophiliac male, X small h, Y. Write down the gametes. Use the Punnett square. Write down your F1 generation genotype, phenotype, and ratio. Here you can see that the males won't pass this disease on to their sons, but they can pass it on to their daughters, who will become carriers of the disease. Okay, let's have a look at what we've learned in this lesson. How is sex determined? Sex is determined by the sex chromosome. When there are two X chromosomes, a female will result, and when there's an X and Y chromosome, a male results. If the X chromosome carries genes that are not in the Y chromosome, these are called sex-linked genes. The allele for color blindness is recessive and is found on the X chromosome. This means that the female is the carrier when heterozygous, whilst the male will be colorblind if the gene is passed on to him, or the allele is passed on to him. Hemophilia is when the blood does not clot. This is because the gene or allele does not function properly. The gene doesn't function properly and the clotting factor 8 is not produced. The allele for this is linked to the X chromosome, so the female is the carrier. Males thus inherit for hemophilia from their mothers, but they can pass the allele on to their daughters. And that concludes our lesson, the end.